Hello, everyone. How are you all doing? I can see we've got 2 people at the moment. Fingers crossed we'll have some more. I've had quite a lot of messages on Instagram saying that you're looking forward to the live stream. So am I. I've been preparing for it, so hopefully it'll be good and really help you with your revision. If you just head over to the comments and have a look, I've asked you just to let me know what you're studying, what course you're doing. So are you studying combined science? Are you studying biology only? And within each one, are you doing foundation or higher? Let me know what course you're taking. So I'm going to give you a little overview of what this live stream is going to cover. So I'm going to split it into 2 parts. The first, we're going to do combined science, because I think more people in the country study that than biology only or triple. So we'll start with combined science, and then we will cover the biology only and triple stuff as well. So with the biology only content, with the combined science content, this is how we're going to split it up. You can see there are 10 main parts to the combined science. And then there's also a required practical for this topic, because we're starting with homeostasis and response. So that's the topic we're studying today. We're going to have another live stream on Wednesday, and that's going to cover the inheritance, variation, and evolution. And then on Friday, we're also going to do ecology. So it'll cover all of your biology paper 2. Okay, and then if you're studying biology only, aka triple, this is what's extra in the homeostasis and response topic. So there are 6 extra little bits you need to know about, and then there's another required practical that you need to know as well. Okay, so, oh, I've had a knock on the door. Bear with me, I'll be 1 minute. Whew. Okay, really sorry about that. I wasn't expecting any visitors. Anyway, let's get going. So we're going to start with the combined science only stuff. So the first part of that is your homeostasis. Yeah. So homeostasis is regulation of the internal environments of both cells and entire organisms. So this can be really important for survival. The main reason why we need this is because everything that lives has enzymes. And enzymes, you'll have learned for paper 1, are these specialized little proteins that help carry out reactions and can speed them up. So that's really important to control. If your, for example, temperature gets too high, the enzymes get denatured. If the pH is wrong, the enzymes might not work either. So we have to maintain a nice constant internal environment. And there are 3 environmental conditions that you need to know for the AQA specification. The first are your blood glucose levels. The second are your water levels. And the third is your body temperature. So those are the 3 examples that you need to know for the AQA specification. Hi, Sala. Okay, and then you also need to know sort of the basics of how this regulation system works, because you can only control systems if you have information about them. So the first thing is that you have got a stimulus. And a stimulus is just any change in either the external or internal environment. For example, it could be your blood glucose levels increasing, or it could be your body temperature decreasing if you find yourself in the Arctic. So that's your stimulus. You'll then need to have receptors of some sort that detect it. There are 5 main sense receptors in your body. I'll just answer that question quickly. We're going to cover the menstrual cycle here, so I'm just going to work through it in order. It's probably going to be about 2 or 3 minutes, maybe more, per section, depending on how big it is. So you can do the math on that. That's when we'll cover menstrual cycle. Okay, so yeah, there are 5 main sense organs in the body. You've got your eyes for detecting light. You've got your nose for detecting chemical smells. You've got your mouth as well for chemical smells. You've got your ears for detecting noise, or sound rather, and then you've got your skin for detecting changes in temperature and pressure, like putting pressure on your body. Okay, so once you've had your receptors pick up that, it then needs to be sent to a coordination center. And you're probably thinking of one coordination center in particular when I mention that. Can anybody tell me an example of a coordination center in the body? You can just type a little comment below. Yeah, very good. The CNS. So that actually includes two coordination centers. That's the brain and the spinal cord. And the pituitary gland is another one. You could also have said the pancreas. Great examples. Okay, so once your coordination center has processed the information, you need to ship it along to have a response. And to do that, you will send it through most likely your nervous system, but it could also be the endocrine system. And then the response is carried out through your effectors. Can anybody tell me the two types of effectors that you have in your body? And Patty, I'm going to cover osmoregulation when I get on to the biology-only stuff. We'll see. Maybe if there aren't a lot of people joining us, then we can move through it. Okay, great. Yeah, we've got muscles, and there's one other type. Great. Well done, Patty. Glands as well. Ellie, well done. Great. So muscles and glands are our effectors, and they carry out the response. That's it. That's your introduction to homeostasis. Number 2, we're going to look at the nervous system. So I've done a little diagram for this one, and I want to see in the comments if you can write down what each one of these numbers represents. So we start with the stimulus, and then there's a chain of events that takes place in your nervous system to then produce a response. So see if you can jot down what 1, 2, 3, 4, and 5, what these pictures represent in this chain. I'll give you all a couple of minutes. I can see some people are quicker off the mark. Yeah, some good responses coming in. Okay, well done, Salah. Well done, Patty. You've got them spot on. I'd love it if we had a second word with the sensory, relay, and motor. What other word? Yeah, Patty, you beat me to it. Very good. So we've got receptor cells, sensing neuron, relay neurons in the middle, motor neuron, this nice big long one, and then you've got your effectors, your muscles, or your glands. I've just picked the pancreas as an example of a gland. Great, so that's our chain of events that happen in the nervous system. Rise with Blue, at the moment I am just doing biology. I've taught chemistry and physics and actually maths as well, all to GCSE. So I am planning on making chemistry and physics videos and live streams, etc. But I've started with biology because it's my specialism and my favorite. So I'm afraid I won't have any videos or content on that for these exams. Oh yeah, okay, Salah, right. Synapses. So now we're going on to reflex actions. So we have got this little gap in the middle, and you're correct, this is a synapse. So synapses are found between neurons. If you zoomed in really, really close on these joinings, you'd see this tiny little gap between them. Can anybody tell me what the purpose of a synapse is? Yeah, they do allow chemicals to diffuse, but why is that important? Okay, so a lot of you know the function, like what they're actually going to do, and they do allow the nervous impulse to be carried on from one neuron to another. But that gap's there on purpose. If you imagine neurons, there are billions just in your brain alone, never mind the rest of your body. If we didn't have that gap and they were all, because lots of neurons synapse onto each other, if you didn't have that gap and the electrical impulse was just able to jump across neurons directly, then actually what you'd see is you'd have huge misfiring of neurons. You'd have basically like a domino effect. If you imagine you have loads of matches beside each other, you light one match, they're touching each other, suddenly the whole thing's going to be on fire. So synapses are actually really important for directing your nervous impulses. So by changing the message from an electrical impulse into a chemical messenger, you allow it to be a little bit more specific. Okay, so that's really great. So lots of you have said that it's going to allow the electrical impulse to pass on. So can you explain what happens in this gap to allow that to occur? I'm going to draw it on in a second. In fact, I'm going to draw it while you're answering. So let's see, how does it actually get across the synapse? What's they? They diffuse. Something is diffusing, but what's they? Ellie, I think you've hit the nail on the head. Yeah, chemicals. So you can call them chemical messengers or you can call them neurotransmitters. Either word or phrase is absolutely fine for AQA. So that's right. They're going to diffuse across the gap from the end of one neuron to the start of the other. They'll just join to little receptors or little sort of holders on the start of the second neuron, and then that will actually generate a new electrical impulse in the second neuron. Okay, so that happens both in the normal nervous system response and in reflex actions, anywhere basically where neurons synapse. But what's the difference? What is a reflex action and how is it different to a normal nervous response? Just dropped my pen. Yeah, so they are faster, and the reason for that is because they actually cut out the conscious part of the brain. Most often they'll go straight to the spinal cord, to the relay neurons there, and then they'll come straight back again. So they actually cut out that extra distance from the spinal cord to the brain and back again. So they're much more quick. Ellie's asked a really good question. When would you mention relay neurons in the sequence of a reflex arc? You would mention them in the exact same place as you would in this chain of events. You'd mention the relay neuron after the sensory neuron. The difference is that these relay neurons, they won't be in the conscious parts of the brain. So like I said, it's probably going to be that it'll go to the spinal cord, and there are relay neurons both in the spinal cord and the brain. And then it'll just come straight back along your motor neuron. Okay, so Salah's mentioned, how about CNS? So the CNS is your central nervous system, which just means the brain and the spinal cord. So the reflex actions, they only use part of your CNS to carry out the response. They sort of cut out the conscious part of the brain and just use the spinal cord. Great. Okay, so the next one on our list for combined science is the endocrine system. So this is our second system in the body to maintain nice optimal conditions, i.e. homeostasis. So can anybody tell me, what is the endocrine system? How does it differ to the nervous system? Really good answers here. Okay, so it is a hormonal system of response, and it acts using glands and hormones and target organs. And because it's using hormones, which are chemical messengers that travel in the blood, this makes it much, much slower. Electrical impulses are really fast. Like, you don't really notice a light turning on. It just seems as though it's either on or off. You don't notice the turning on. But with a hormonal response, it's a lot slower because we have to wait for those chemicals to get pumped around our body in the blood system. So your glands will release hormones. They travel in the blood to a target organ. And there are some examples that we're going to look at. But first, we're going to look at the 6 glands and their placement in the body that you need to know. So have a look. Can you write down what each number stands for from 1 to 6? So you have to memorize these ones. You could get a nice question. I think it would be a nice one if you were just asked to identify a gland on the body. That would be a good one. Nice support going on there in the comments. Please feel free to join in. I can see we've got some quiet watchers, but you should have a go. Even if you don't want to do it in the comments, grab some paper and pen and see what you can write down. It'd be really good to test yourself. If you don't want to see everybody else's answers before you've had a chance to do yours, I think you can just close the comment section as well. Okay, so 1 is the pituitary gland, 2 is your thyroid gland in your throat, 3 is your pancreas, 4 are your adrenal glands, they're just above the kidneys, 5 are the testes on the male body, and 6 are the ovaries in the female body. Okay, so those are the 6 glands that you need to know in the body. One of these glands is a master gland. Can you tell me which gland it is and what we mean when we say master gland? I'm just writing the names on while you answer that. So which one is the master gland and what do we mean when we say master gland? Okay, some good answers here. So Sala says it releases all hormones, while Patty says it can release multiple hormones. I'd like to go with Patty's answer. It's just slightly more specific in that it can release multiple hormones, and basically it can coordinate effects across the body because of that. So we're going to look at one example now. If you're studying higher tier, we are going to know about thyroxine and adrenaline. Okay, so thyroxine, its levels in the body are actually controlled by both the thyroid gland and the pituitary gland. So that's going to be one example. And we'll start by talking about what the thyroid gland is for. What thyroxine is for. This isn't a spec, it's kind of hidden. Maybe you haven't heard about it a lot, but do you know what thyroxine does in the body or what it controls? Very good. Okay, so it controls growth and development in children, and then in adults, it controls your basal metabolic rate. Very good. So that's thyroxine's function, and it's released by the thyroid gland, which makes sense. They sound pretty similar. Okay, then we've got adrenaline. Okay, where do you think adrenaline is released from? Again, it sounds pretty similar in the word. Yeah, it's the adrenal gland. Very good. Okay, and adrenaline, it impacts your bodies in lots of ways. It increases your heart rate. It increases the supply of oxygen and glucose to both your brain and your muscles. And your breathing rate, you'll start to breathe really, really fast and start to feel all nervous. Basically, it's your fight or flight response kicking into gear. So if you encounter a scary situation, you're probably going to release some adrenaline pretty quickly. Okay, so we're going to now look at the example of thyroxine within negative feedback. So again, negative feedback is something that is only for higher tier course students. So you could be asked about an example that is in the specification, or you could get a completely different example that's brand new, but you just have to interpret it using what you know about negative feedback. So essentially, negative feedback is a loop that will try and return your conditions to the set point. And the set point is just whatever is normal. So for example, for body temperature, it would be around 37 degrees Celsius in the human body. And you'd have feedback systems working if it goes above that, and feedback systems working if it goes below that. So we've got the example of thyroxine here, and we'll start by looking at the purple route. So let's say it deviates from the set point by increasing. So your thyroxine levels have gone too high. So the response is going to be that the pituitary gland detects this, and instead it's going to release less TSH. And TSH stands for thyroid stimulating hormone. So here's an example of how the pituitary gland is a master gland. This is one hormone that's then going to travel and affect another gland. Okay, so less thyroid stimulating hormone means that when it reaches the thyroid gland will release less thyroxine. Makes sense. And by reducing the amount of thyroxine produced, you will eventually return to the set point. But the reverse can happen as well. So I want you to tell me what the alternative fruit of negative feedback would be within this example. Take a look at it and see if you can write down what 1, 2, and 3 will be. I'll hold it a bit closer. Any answers? I can see Salah saying decrease, but what would number 1 say? Yeah, okay, so that's a great start. So first of all, the thyroxine levels will decrease. So it's the opposite to this one. This time they increased. So in the pink pathway, it would be the thyroxine levels decrease. That's a good start. And then what's going to be the next step? What will detect that decrease? Yeah, it's going to be the pituitary gland. Again, it's going to be the same one. And instead of releasing lots of TSH, instead of not releasing lots of TSH, it's going to increase its TSH production. And when it increases the thyroid stimulating hormone, then the thyroxine levels will increase because the thyroid gland makes more of them. Okay. All right, let's go on to our next one. So, control of blood sugar. This is sort of a two-part one because if you're foundation, you'll only learn about high blood sugar, but if you're doing higher tier, you'll need to know about low blood sugar as well. I'll just write a little H beside that. Okay, so this is a nice chain of events that happens. So first of all, your blood glucose levels rise whenever you've had something like a sugary meal or a meal that's really high in carbohydrates. Hopefully from your paper 1 revision, you'll have seen that carbohydrates are broken down by amylase into glucose or sugar. So that increases your blood sugar levels. Okay, so we've got a little chain of events here. The first step is that increase in blood levels are detected by the pancreas. So the pancreas, remember, is a gland, and glands can produce hormones. So we've got a little hormone here, represented by the purple, that's being produced for high blood sugar. Can you tell me what you think that hormone is? Thanks, Patty. Appreciate that. I'm not an art teacher. Not even taking it for GCSE myself. Very good. It's insulin. Okay, so insulin is produced, and you can see it then is going to go on a little journey, and it's going to be carried in the bloodstream. And you'll see this glucose is also being carried in the bloodstream and just some blood cells. Okay, so the glucose it can go into 3 different places with the help of the insulin. And let's see, can you tell me what each of these 3 places are? Good. Muscles, liver, and this one? What do we think this one could be? Don't know. Well, it's probably confusing because none of these are done to scale, but this is just meant to be a cell. Yeah, thanks, Carl. It is a cell. There's your nucleus, some mitochondria, some ribosomes if you look really close. Yeah, okay, so with the help of insulin, your glucose can be taken up by your liver, your muscles, and your cells, just general body cells, for respiration. But when it's taken up into both the liver and the muscles, something special happens. Does anybody know what happens with excess glucose, what it is turned into? Very good. I would just like you to check your spelling there. Salad, because examiners want it letter for letter perfect. If you mix the words glycogen, glucose, and glucagon up, then you're going to lose your marks. So just have another go. Yeah, perfect, glycogen. Okay, so in both the muscles and the liver, excess glucose is converted to glycogen. I'm just writing that down so I can put it up on the screen. Glycogen. Helps if you say it out loud, I think. There we go. Okay, so that's all you need to know if you're studying foundation. But if you're studying higher tier, you need to know what happens if you've got low blood glucose levels. And this could happen due to exercise or just having not eaten for a while. Your cells are using it up, respiring. So we have another little chain of events happening here. You were great at this one. Let's see, can you just give me a lovely, clear sentence to explain the chain of events that takes place when you have low blood glucose sugars or levels? While you're having a go at explaining, I'm going to write it down. And again, see if you can be really careful with your spellings here. All done? That's a really, really good start, Patty. I'd like to hear the rest of that. Sally, you've kind of got the latter part of it, but that's good. Okay. It's quite a long sentence. If you do it all in one sentence, it's pretty long. Yeah, really good. Okay, so it's hard reading this. It's back to front for me when I look at it on the screen. Pancreas detects the low blood glucose and releases glucagon. Check your spelling, that has to be spot on. Which then travels in the bloodstream to the liver, which is then causing it to convert glycogen back into glucose, which is released into the blood. That last part of it being released into the blood is often forgotten. It is important because we're talking about the blood glucose level. If it just turns it into glucose and it just stays in the liver, that doesn't actually restore our blood glucose levels back to normal. So just make sure you don't forget that. Okay, so with that in mind, you then have to know about your type 1 and your type 2 diabetes. So you could get a nice comparison question on this. You could get some graphical data to do with this. But we'll just very quickly go over what the disorder is, what causes it, and how you treat it. So type 1 diabetes is when your pancreas doesn't produce insulin in the first place. And the cause of this is normally genetic. It's something you're born with, and it will develop when you're a child. So the treatment, because your body's not producing the insulin itself, is you need to get the insulin into it. And through the wonders of genetic engineering, we've been able to harvest huge quantities of insulin from modified bacterial cells. And diabetics with type 1 diabetes will just inject themselves with insulin before a meal. And they need to, of course, monitor their blood glucose levels really carefully throughout the day. They'll also try and monitor it and control it through the use of exercise and diet. So that'll be another part of their treatment plan. There's loads of great information on the NHS website about diabetes. And it's really important to know about it. Type 2 diabetes. So type 2 diabetes is one that you develop during your life, and it's often due to obesity. So that means having a very high BMI. It's due to old age. You're more likely to develop it as you get older and older. And it's due to a lack of exercise. So all three of those are your risk factors for type 2 diabetes. Okay, the treatment then is slightly different for type 2 diabetes. Your body is still producing the insulin, but the issue is that your cells are not responding to the insulin. So the liver, it's not saying, oh, I need to take this extra glucose in and turn it into glycogen. It's not doing that. So the blood sugars are going too high. So injecting more insulin doesn't often work. So instead, you need to control it through more exercise, which makes sense because that was one of the risk factors that caused it. Losing weight and doing that by controlling your diet so that you've got a lack of carbs or just the right amount of carbs and sugars. There are certain drugs that can also help to treat type 2 diabetes. Okay, that one, it could come up. It's a big problem in our developing world. Unfortunately, our diets are getting worse and worse, and we have too many processed sugars and unhealthy things in our foods. So that is a disease that's on the rise, and I think maybe exam boards want to make sure you know about it and how to prevent it. So there you go. Oh, it did, Patty. I didn't hear that. How did it come up in paper 1 if it's a paper 2 topic? Maybe it was some sort of data question. I heard there was a lot of maths on that one. Okay, well, we're not far off the end of the combined science. Number 7 is reproductive hormones. So there are one big male reproductive hormone and one big female reproductive hormone that you need to know. And these cause secondary sex characteristics, which are the changes in the body that take place generally during puberty. So what is the male reproductive hormone and what is the main female reproductive hormone? See if you can tell me in the comments. Good, Carol. Testosterone is the male reproductive hormone. And what's the female one? Sala, the ovaries are where it's produced. Let's see if we can pinpoint the name of it. Yeah, estrogen. Okay, so we need to zoom in a little bit more. Oh, and yeah, you do need to know where they are made. Well, so testosterone is made in the testes, and estrogen is made in the ovaries. Okay, we need to zoom in on 4 of the female reproductive hormones that are specifically for regulating the menstrual cycle. Now, there's sort of 2 levels to the menstrual cycle that you need to know. If you're foundation, you need to know the names of these 4 hormones, but you only have a little bit to learn about them. If you're doing higher, then we're going to do another section on the menstrual cycle that's going to go into quite a bit more detail on each one. Okay, so see if you can tell me what each one of these hormones is. I've told you what it does, but I've left the name blank. Oh, Sala, you're very kind. Well, I'm here to teach kind of online, so I guess in a sense you have got a teacher like me. Yeah. Any ideas? 1, 2, 3, 4. What is each one of these? We've gone a little bit more quiet. Maybe this is a harder one. I know there's a wee bit of a lag. There used to be, well, there was the last time I did my first ever live stream, there was a lag. Yay, we have an answer. Okay, and let's see if you're right, Patty. Yeah, you're spot on. And 3 and 4 could be the other way around because I've said both of these do this. So, first one is FSH. It causes an egg to mature. How often does that happen, do we think? Sala, you must be doing higher tier. You've already started going on to that sort of level of stuff. That's very good. So it causes an egg to mature once every 28 days or once every full menstrual cycle. And FSH just stands for follicle stimulating hormone because the follicle is where the eggs develop. Great. The next one is it causes ovulation, and that was LH or luteinizing hormone. And the third and fourth one, they both cause the same thing to happen, which is maintaining the uterus lining, i.e. keeping it thick. So that's estrogen and progesterone. Just while we've mentioned LH and ovulation, do we know when ovulation occurs? So yeah, ovulation happens on day 14 of the menstrual cycle. Okay, so if you're now studying higher tier, we're going to go into the more detail that you require for this. It's really likely that you could get a data question with this because it is, it's one of those ones where we've got different levels of hormones throughout the month. So it's really likely you're going to get some form of graph question. Okay, so I've written them down because there's a lot and I'm slow when I write, and I know that's a bit boring for a live stream. So I've written down what happens for each one, but we're going to go through it on the diagram just to try and help it make sense for you. So I've slightly changed the order, and that's because there is a little bit of a logical progression of how each hormone impacts the next hormone. So we'll start with FSH. So FSH is produced by the pituitary gland. There's another example of a hormone that this master gland makes. And it causes an egg to mature. We just learned that in the previous foundation level stuff. So what we've got is one of our little eggs in our ovaries maturing. I'm just doing it like that. And then estrogen, it causes the lining of the uterus to start to develop. So our uterus is this main part here. And when the lining develops, it means it gets thicker. So it gets like a blood supply, a good blood supply coming to it. And the walls, they're getting ready in case the egg gets fertilized. So they're getting ready for implantation. Sorry, I think did I say that? It stimulates estrogen production. That's why I've done estrogen next. So it stimulates estrogen to be produced in the ovaries, like we just saw. Okay, so our lining gets nice and thick, and then we don't want any more eggs to mature. You only want one egg a month to mature and be released during ovulation. So estrogen is going to inhibit FSH, which just means it stops the production of FSH. So it kind of doesn't work very well when I try and write with my hand as a back support, or else my pen's running out. No, it's good. So it inhibits or stops FSH, and then it stimulates LH production. So LH is our luteinizing hormone, and that causes ovulation on day 14. So the next step is ovulation, which is our egg traveling out of the ovary and into the oviduct, where it may or may not meet a sperm. Okay, then we've got progesterone. So progesterone is produced by the empty follicle. If you remember, I said the follicle, the F for follicle, is where the egg is produced. But once the egg leaves it, it's empty, and that makes progesterone. So progesterone then inhibits, which again means it stops the production of LH and FSH. So we don't want any more ovulation, and we don't want any more mature eggs. So I'll draw some arrows on that to show you that it stops both of those from happening. So it all kind of overlaps. Finally, progesterone is responsible for maintaining the lining of the uterus. And that means it keeps it nice and thick. Fertilization, there's a little window for fertilization to happen. The sperm can survive for a few days once it enters the female reproductive system. So it swims along here and it might fertilize the egg. And of course, the egg then has to travel from the oviduct down to the uterus to implant. So you need it to stay thick. So that lining, for about 10 days, it will stay thick, ready for a fertilized egg to implant. If it doesn't get that egg coming in, then it will just break down and it will leave as a period. So that's our menstrual cycle for higher tier. I think maybe it was Patty, I can't remember, Somebody said they were struggling with the menstrual cycle. Does that cover it, or is there anything more that you would like to know about it? Let me know. It was you, Salah. Well, let me know. Little more. It covers it a little more, or you're still stuck? You need a little more help? Patty, corpus luteum. I don't know what that is. No idea. It's not on the AQA spec. It's not on the A-level spec. As a woman, I don't know what it means. Maybe you can tell me. But you don't need to know it for your GCSE. So, Sally, you said you're not fully understanding. Well, it's kind of one of those things that you're going to have to practice a few times. What builds up? So the best way I can think of it is you write down each of your hormones and then you practice just covering up your notes, or my notes if that's what you're using to look at, and you just write down what each one does. And then you uncover it and you see how many you got right, and then you practice it again for the bits you got wrong. It's definitely like a hard part. I'd say it's one of the hardest parts of this topic, just because it's a little bit abstract with those letters, and yeah, it's a long process that's going on. When the ovum has no sperm to form a zygote. I'm still not sure what it means. Is that a noun? Corpus luteum. I'll have to look it up. Corpus luteum. Let's see. A hormone secreting structure that develops in an ovary after an ovum has been discharged, but degenerates after a few days unless pregnancy has begun. So it's going to secrete a hormone that probably helps with pregnancy then? Is that right? Here we go. People also ask, what is the function of the corpus luteum? For establishing and maintaining pregnancy in females. And it secretes progesterone. Of course, that makes a lot of sense. You don't want your progesterone just to maintain the lining for 10 days. You want it to maintain it for the whole pregnancy if you get pregnant. So that's really good. I've learned something new there, Patty. Thank you. Salah, that's kind of you to think that you've learned a lot, but remember, it's testing yourself that helps you learn, and I'm sure you've learned absolutely tons. Not everything you learn in school is content. You learn a lot of social things as well. But thank you. Okay, let's move on. We're going to go on to number 9, which is contraception. So on the specification, the big part of contraception is evaluating them, which basically means you're going to be given some information. You're not going to have to do all of these, memorize all of them yourself. So expect some information. So what we're going to go over is just very brief. So you have an overview of the different methods, but you don't have to worry too much about memorizing them. Thanks, Patty. Good luck for your exam. Appreciate you joining us. Okay, so hormonal control of pregnancy, or preventing it, starts with a pill. This will inhibit FSH. As we just saw in the previous bit of information, there are 2 hormones that inhibit FSH. One is progesterone and one is estrogen. So the pill can be using progesterone or, that's the wrong way up, progesterone or estrogen, or actually a combination of both. So they'll both inhibit FSH, and that stops eggs maturing. You can get pregnant if there's no mature egg being released each month. You also then have the patch injections and the implants, which is just a little, I think it's made of, not actually sure, plastic? I think, not sure, I actually have to check. You get that injected normally into the arm, and it will slowly release progesterone. So these methods are all a little bit longer lasting, particularly the implant. The injection, you'll need it every few weeks. The patch can be a daily thing, sometimes a weekly patch, but they all use progesterone as well. Then we've got the non-hormonal methods. starting with spermicide. That is chemical. So it's essentially killing the sperm. That's very ineffective. It should only really be used in combination with another method. You've got a high risk of pregnancy with that one. Then you've got your biro methods, such as condoms for males and diaphragms for females. And those both prevent sperm from entering the female reproductive system. Their big advantage with those is that they can help prevent some STDs, not all, but some it will help. And the disadvantage is that sometimes they're not fitted correctly, and that can cause a risk of pregnancy if they're damaged or they break, for example. Next one is IUD, and that stands for intrauterine device. So this is something that would be inserted into a woman's uterus by her doctor. And essentially it will stay there for as long as she wants, but then she'll have to revisit and have an appointment to get it removed professionally by another doctor. And it works by preventing the implantation of an early embryo. So that might be against some religions because the embryo has been created and it's the implantation that it prevents. Some of them will also release progesterone. So there's a tiny bit of crossover there. You can get some that release progesterone. The next one then is surgery. So this is a permanent form of contraception. It can be done with both males and females. Essentially, you will tie a little knot or actually cut either the ovaries or the sperm ducts in males. So it's pretty permanent. I think you can undo it, but it's not a certainty. So you should only do it if you're pretty certain that you don't want to have any more children. And then finally is abstinence. Abstinence is refraining from having sex around the period of ovulation. So working out when you're going to have an egg that could be fertilized and then not having sex on those days where there's a risk of fertilization. That's a very risky strategy. You can definitely get a high risk of pregnancy with that, as it's very hard to predict. So yeah, that one's not great. But of course, there's no side effects. So side effects are something to think about for these. Of course, where there's hormonal control, there can be side effects. You need to think about how easy it is, how expensive it is. The biggest thing is how effective it is. Does it prevent pregnancy or not? Okay, and then the last one for combined science is our infertility treatments. So we've got a little diagram here of how we can treat infertility using IVF, in vitro fertilization. And we'll just first of all talk briefly about what infertility is. It's the inability for a couple to get pregnant. So this can be a problem with the male reproductive system. Maybe he's got a low sperm count, or maybe there's a defect with the sperm. For example, its tail not functioning properly, so it's not really able to swim to meet the egg. Or it can be a problem with the female reproductive system. For example, a blocked ovary or damaged ovary. A lot of them is just that we don't know. Doctors, sometimes they're stumped. They can't work out why a couple can't get pregnant. It's actually pretty common. The NHS recommends. They've worked out it's something about 1 in 7 couples struggle to get pregnant. So it can be a lot harder than it appears sometimes. Scarred tubes, yep, that's another possibility, Carl. Okay, so IVF is one treatment for infertility that you can try. It's a pretty tough process. It takes a lot out of the couple, particularly the woman. But if it results in them having a child that they really want, then ultimately that can be a really nice and happy thing. So you need to know the stages of IVF. See if you can tell me what is happening in number 1. Yeah, Salah, I love the idea of adoption too. I think it's really nice to consider adopting a child rather than having one of your own. Got a lot of people on this planet But of course, it's also nice to have your own children too. It's just a personal decision, isn't it? Anyway, what's happening there in number 1 of this IVF cycle? Infected sperm in the womb, or maybe injected, you mean? Well, that would be very direct. It's a bit of a longer process than that, I'm afraid. So number 1 is actually, that's a woman we've got there, and she's been injected with fertility drugs. So it could be that she's not making enough of those hormones that we just mentioned. So FSH or LH. Well done, Ellie. That's exactly right, fertility drugs. Number 2 then, once we have given her the FSH and her eggs are maturing, we can then extract them. So number 2 is showing the extraction of the mature eggs from the ovary. Number 3, the eggs are then put into something like a Petri dish in the lab, and they are mixed with sperm. This is not to scale at all. The eggs are a lot bigger than the sperm. But anyway, so they're put together and hopefully fertilization will occur. So the sperm and the egg will fuse together. This needs to be monitored. So number 4 is that we are going to use microscopes, which is a great advancement. They've got a lot better in recent years. And we're going to check that, we're going to put this under the microscope. And we're going to check that the early embryos are developing. Some of them won't. Not every egg and sperm that fuse actually develop and become a viable embryo. So you need to check that they're developing at the right rate, producing the right number of cells, etc. So then you get your early embryos, which are just these balls of cells. And in number 5, you're going to choose 1 or 2 to insert. I don't know why I did my arrows there. That's completely wrong. That's me rushing these diagrams. You don't want to insert them in the ovary. That's not good. You've got to insert them into the womb. Let me use my purple pen to redo that. There we go. So you need to put your embryos into the womb. So I've noticed that Salah has said, doesn't it lead to multiple kids? And yes, it can. So when IVF first became an option, they used to insert more than 2 embryos, and they had a very high incidence of triplets. I don't know, maybe even more than that. Triplets and even twins, they're more dangerous for both the woman and the babies. The babies often have a lower birth weight, which can lead to complications, and can be a harder pregnancy for the mother. So that's why they now reduce it and they only put in 1 or 2. So yeah, that's really a good point. Well done. Okay, so those are the stages of IVF. And that is the last topic within infertility treatments and last bit of homeostasis and response for combined science. Does anybody here study higher tier? Not higher tier, study biology only, or triple, as some people call it? Yes, we do. A couple of us. Okay. Would you like to also go over the triple only content? I will put that up here. Yes and yes, we would. Okay, so this is what we've got for triple only or biology only content. Okay, so I'm afraid I ran out of time on doing my nice diagrams. I do have some diagrams in another notebook. They're a little bit more scribbly, but we can go through the ones that I have done, and I'll just talk you through the bits that I haven't, or I can just draw them right now. Okay, so number 1 is the brain. So there are 3 parts of the brain that you need to know, and you need to be able to draw them on a diagram. So you've got your cerebral cortex, you've got the cerebellum, and you've got the medulla. I'm going to sketch a quick diagram, and let's see if you can tell me which one is which. And I've also got a little hint to try and help you remember them the way that I remember them. This diagram is going to be pretty bad, I'm afraid, because I'm just doing it right now. So, 1, 2, 3. So, which one is the medulla? which one's the cerebral cortex, and which one is the cerebellum? My tea's probably cold, but I'm going to drink it anyway. I don't know if there's a lag or if we've just gone quiet. Do we know which one is which? Ellie does. Okay, 1 is cerebral cortex, 2 is cerebellum, and 3 is medulla. You are spot on. So I'll tell you my way of remembering it. Number 1 is the cerebral cortex, and I remember it because it's the longest word, cerebral cortex. There's 2 parts to it. So it's the biggest or the largest part of the brain. Then medulla starts with an M, M for middle, and it's like in the middle of the cerebral cortex. If you line it up, kind of cuts it in half. And then the cerebellum, number 3, that's just whatever is left behind. So that's my trick for remembering the parts of the brain. You then need to know what each part of the brain does. So this is one of those ones where it's a little bit of abstract information. It can be very easy to mix it up. So I recommend you come up with a mnemonic, and I have come up with a mnemonic. Let me see if I can find it. I should remember it, to be honest, if it's a good mnemonic. What did I come up with? Oh yeah, chickens can cause memory loss. Could maybe be messed up antibiotics. Yeah. Okay, so this is my mnemonic. Chickens could cause memory loss, could maybe be messed up antibiotics. So I'm thinking of some chicken hypnotizing you, causing memory loss because they've been given messed up antibiotics. I don't know. But I'm going to tell you what this stands for, because this is going to tell you what the parts of the brains do. So the first bit, the CC, is cerebral cortex. And that is consciousness, memory, and the L is for language. C is for cerebellum. And the M and the B is for muscles or muscular activity. And the B is for balance. And the last one is medulla. And UA is unconscious activities, i.e. things like breathing, control of heart rate, etc. So that's what you need to know. To me, that is kind of abstract. Unless you study psychology, and you've probably seen it enough times that you know about it. But mnemonics, they are a great way to try and remember some abstract information. It worked for me. Maybe it'll work for you. But maybe come up with your own mnemonic, something that you're going to find easy to remember. I always recommend that you put a person's name into your mnemonic, especially somebody that you know is going to be sitting near you in an exam, because it can act as a nice little memory trigger. You see them and you're like, oh yeah, I had that weird mnemonic about that person. Yeah, and that can help. Let me know what you think of that one. Okay, so that's the first bit of the brain that you need to know. Let me see, is there anything else on the brain? Let me just scroll through specification. I haven't done my videos on them, so I'm not just as hot on remembering everything you need to know. Oh yeah, so you should be able to remember how we can know the functions of the brain, so how scientists have worked this out, and also some of the difficulties with treating brain disorders. Sorry, bit of reverb there. We can discover brain disorders, we can discover the functions of the brain using MRI scans, which is where a person is put in this big scanner and you can see what they're doing. So maybe they're reading a book and you see parts of the brain light up in the MRI scan, and that means you know those parts of the brain are involved with processing written information. So that's one way. The second way is studying people who have got problems with their brain. So let's say you know that they have a damaged medulla and they have breathing problems. Maybe that means the two are linked. There's a classic example of Phineas Gage. He was this guy, I think, in the 18th century, and he got a big metal pole stuck through his brain, and he survived, but he lost all of his social inhibitions. He started gambling, spending all his money in bad ways. He used to be really polite, hardworking, but his personality completely changed. And so they discovered centuries later that that part of the brain is responsible for sort of your social cues. Okay, so that's the second part. And the third part is by electrical stimulation. So if you have cut open part of the brain during surgery, you've cut open the skull to reveal the brain, and then you electrically stimulate parts of the brain, and the person experiences something like pain, then you know that part of the brain is responsible for that. So that's how you can discover what the parts of the brain do. And then the last bit is difficulty in treating the brain. If you have a brain disorder, it can be really easy to damage the brain unintentionally. So surgery is often done with the patient still awake, so they can tell that nothing bad is happening. And the brain's just really complex. We have not worked out how it all works yet, so it's hard to treat brain disorders. Okay, so that's the brain. Ryan, sorry if I'm saying your name wrong. Can I do the water cycle? I can, but I think that comes up in the ecology topic, and I'm going to do a live stream on that on Friday. So you can check back in on Friday for that. Right, need to go back to the front. Next one for biology only is the eye. Okay, the eye is, I like the eye. It's kind of physics, to be honest. Lenses. So if you like physics, you might like the eye. Here's one of my diagrams, my less nice ones. This is showing you the parts of the eye and what lots of them do. So I'll just go through different parts. So right at the front, you've got the cornea, which is sort of a hard protective layer. It's part of the sclera, which goes around the whole of the eye, but the cornea is just the part at the front. So it's, as well as being protective, it's the first bit that light hits when it enters your eye. And so it causes the light to refract, which means it changes direction and sort of funnels it into the eye. Behind that, you've got the iris. The iris is the colored part of your eye, and it can change in size. So if it opens up really wide, then your pupil gets a lot bigger. Or if it sort of comes really small, then you'll have a really small pupil. And the purpose of it is to then restrict or control how much light is getting into the eye. Too much light in the eye can damage the light-sensitive cells of the retina, which is at the back of the eye. So you don't want to let too much light in. But you need enough, obviously, to see. Behind that, then, is the lens. The lens is for the fine focusing of light. So we said that the cornea, it refracts the light, and then the lens will refract it a little bit more to perfectly place it on the back of the eye, on the retina. So you always want the light to meet where the retina is. If it meets in front or behind of the retina, then you're going to have a short or long-sightedness and the image will be blurry. Okay, so the lens can change shape because it's, sorry, a bit noise there. It can change shape because it's kind of like a jelly-filled sac. And the two things that help do that are the ciliary muscles and the suspensory ligaments. So the ciliary muscles are the ones at the top, and the suspensory ligaments are attached to those. And they can just be taut, which is when they're pulled, like stretched out, or they can be loose. So they're like a bit saggier, I guess. And that changes the shape of the lens. You've got a blind spot at the back of the eye, which is where the optic nerve joins to the eye. You need to obviously get this message from your eye to the brain to actually process what you're seeing. So the optic nerve does that, but it creates this little blind spot. Now, you don't normally see it because your mind fills in the blank. Your brain sort of just, I don't know, makes a collage of all the input and sort of glosses over it. And I think that is it. The retina, that's just the back of the eye that's got those photosensitive cells. Okay, so that's the structure of the eye and how each part functions, what it's for. But you also need to know what problems the eye can have. So there's hyperopia and myopia, which is on the other side of this. So myopia is short-sightedness. The way I remember that is the my part is like I can see myself. So that's myopia. Hyperopia is so myopia, you can see short-sighted stuff. You can see yourself, so you see short-sighted stuff, but something in the distance, really far away, it would look blurry. And the reason for that is because your eyeball is either too long, or maybe you've got a really short fat lens. So the light is coming in, will focus in front of your retina. So it's focusing too early. Hyperopia is the opposite. So this is long-sightedness. So you have no trouble seeing things that are really far away, but maybe if I tried to read this and I had hyperopia, it would be all blurry because I'm long-sighted. Okay, so this is the opposite. You have got an eyeball that is maybe too short, or you've got really long, thin lens, and so the light that's coming in would focus behind the eyeball. So it focuses too far away. You need to know how you correct each one of these. So for myopia, you can use a concave lens, and that will basically just spread out the light a little bit more before it comes into the eye. And then when it gets refracted, it will refract to the back of the eye. Hyperopia, you need to use a convex lens, which is one that looks like this, and that will help divert the light closer together. Okay, so that's the eye covered. Let's see, what else have we got? Control of body temperature. As there's not many of us left, I'm just going to show you it in the comments. If you would like to go through any of the rest of these, let me know. If not, I'll just answer any last questions and answers, and then we'll tie it up, as I have gone a little bit longer than I expected to again. So let me know, is there anything else you want to do, or shall we just tie it up there? Ellie, you're interested in the plant hormones. Yeah, that's nice and brief, so we can go through that. That's no problem. Okay, so plant hormones, their big one you need to know about is auxin. So auxin controls the growth of roots and shoots in response to both light and gravity. So shoots obviously want to grow vertically, and they want to grow towards the light. So auxin in shoots causes cells to elongate or grow longer. For AQA, they're actually not too picky on you knowing the details. What they want you to know is that it causes unequal cell growth on each side of the shoot. So I'll do a little drawing. I will explain the science in a bit more detail, but if you don't understand it, you can just learn that auxin causes unequal growth on each side. Okay, so let's say we've got a shoot like this, and we've got some sunlight. Before the sun was there, the auxin would be evenly distributed. So your hormone auxin would be all over the plant. But when the light comes along, the auxin will move to the shady side. This side would be in the shade. And you'll have more auxin here. And more auxin means that the cells on the shaded side will grow longer, and that causes them to curve. So you've got a sort of curving towards the light, like this. The same thing actually happens for gravity in shoots. Auxin will still cause the cells to grow longer. So let's say we've got a newly germinated seed, and we put it on its side. Auxin, it will collect on the bottom side. It will do that in both roots and shoots. So I'll do them both on the same diagram. This isn't a very good diagram. So the auxin will collect on the bottom of the shoot. This is our seed that it's just hatched out of. I'm not sure if hatched is the right word. So the auxin will collect on the bottom. And remember, in shoots, it makes the cells elongate. So these cells will grow longer, and that causes it to grow up the ways or vertically against gravity. So shoots always want to grow against gravity. But auxin does something different in roots. Roots obviously want to grow towards gravity. So it actually inhibits the growth of the cells in the roots, which means it sort of slows them down. So these cells, they stop growing as much. So the other cells, because they're growing at a faster rate, they will start to curve down the ways. So if you imagine your root is in some soil, that means the root will grow down towards gravity and the shoot will grow up away from gravity. It's hard to draw like this. Okay, so that's what you need to know is essentially that auxin causes unequal cell growth in roots and shoots. Okay, then there are some other hormones that you need to know about. So auxin stimulates growth in shoots and inhibits growth in roots. Yep, that's spot on, Ellie. That's right. And the faster it grows, the longer the cells. Yes, correct. I think you've got it. And hello again, Salah. Nice to have you back with us. Okay, would we have to link that to geotrophism and phototropism? Ellie, are you studying AQA? Yes, you are. So let me just double check. Those are the words which mean response to light and response to gravity. So it's just whether or not you need to use those words or not. Okay, so I've got the specification up. And actually the words are there, but they're in brackets, which mean that you don't have to use those words. So it just says that blah, 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 responses to light, brackets, phototrophism, and gravity, brackets, gravitrophism or geotrophism. So it doesn't matter if you use those words or if you explain it's a response to light, it's a response to gravity. It's absolutely fine. But what I've just described, this is phototrophism and this is geotrophism, response to gravity. Okay. Do you want to know a little bit about the other hormones, like gibberellins and ethine, and a bit more about the other uses of auxin? I think that's higher tier for biology only. Yeah, I've got a little bit of time. I have some friends coming over soon, but we've got some time right now. Okay, so we'll go through that. So ethine, you might have heard about that in chemistry, maybe. Let me just double check. So that's for ripening food. So let's say we get a lot of foods exported these days. Let's say you've got a tropical fruit coming from another country to the UK. You obviously don't want it to ripen before it arrives. So you'll pick them early, you'll transport them, and then when they get to the shops, you'll use ethine, you'll spray it over them, and then they'll ripen ready for you to buy and eat. So that's ethine. Next is gibberellins. They've got a few things that they do. First is that they can increase the fruit size. Plants will naturally produce gibberellins, and we've sort of harvested it and made synthetic versions of this hormone. So, for example, you can buy seedless grapes, and then the seeds have been removed. They've been modified to grow without seeds, but seeds normally contain the gibberellins to make the grapes grow big. So seedless grapes are normally really small. So what we've done is spray them with synthetic gibberellins, and then the seeds grow nice and big again, so you can enjoy seedless grapes. So that's another thing. The second thing then that they do is they end seed dormancy. So seeds like this, they'll only grow if they've got the right conditions to grow. And sometimes they don't germinate. They don't, grow into their plant unless they have this trigger from the gibberellins. So they can be used to end seed dormancy. And it does mention briefly that that is for malting, which is like in the brewery process. And then finally, gibberellins are used to promote flowering. So a lot of flowers, or a lot of plants, will only flower at certain points of the year. You'll have noticed that a lot living in the UK. For example, springtime, you get lots of bluebells. But sometimes people want to have flowers at different times of the year. So you can use gibberellins to artificially make the plants produce their flowers out of season. Okay, and then finally, auxins. We've mentioned, of course, that they can help cells grow longer, and that growth aspect is used for them. So there's a funny way that they're used. It sounds counterintuitive, but they're used as weed killers. Now you might think, what? That's crazy. If I spray some weeds with auxins, they're just going to grow. And you think, oh, that's bad. But actually, the thing is that it causes them to grow out of control, kind of like cancer in the human body. So when the plants grow out of control, they actually die. They can't get enough nutrients, etc., to support that rapid growth. So they die. So that's the first thing. Auxins are used as weed killers. They're also used as rooting powders. So when you take a plant cutting, let's say you've got a rare plant, you can snip off a part of it and you'll dip it in some auxin as a rooting powder, and that will encourage new roots to grow so that when you plant that cutting, it actually works and will grow a brand new plant. And then the third thing is for growth in tissue culture. That's really similar. Tissue culture is when you're growing a new plant, more often in the lab, and you can just use auxins to help promote the growth in the tissue culture. Okay, so that's everything on plant hormones. I did spot a couple of questions there. Sala, I'm afraid I have no idea what you mean by that question. And then, I'm not going to church. I don't know what you're saying. Carol, what other subjects do I do? Well, I mean, I don't do any subjects anymore. I've finished studying, but what other subjects I teach? I have taught physics and chemistry to GCSE, as well as maths, and I teach biology at A level, so that's my subjects. But I like studying, I like learning about lots of other subjects. Sally, I noticed you also earlier said, is combined harder than triple? I'd say that they're of equal difficulty. They both go up to grade 9. You both have the capacity to be tested at that really high level. Obviously, there's more content for triple, but you should have had more time in your school year to learn it. So they should really be equal. It's just your preference, whatever one you've chosen to study. Okay. Are there any last questions, or shall we wrap up there? I think we covered, pretty much everything. Not all of biology, but I think, yeah, you can take a little look at that yourself if there's anything else you want to know. Please subscribe if you haven't already. Check out my other videos. I have got an entire Paper 2 biology video that's live. So check that out. I've also got a math and biology video that is nearly done. I just need to edit it and then it will be up hopefully by tomorrow. And I'm going to do more live streams. We'll be back on Wednesday, same time, 5 o'clock for about 1 hour, hopefully maximum. And we'll be covering inheritance, variation, and evolution. That's topic 6 on paper 2. And on Friday, we'll cover ecology, same time, 5 o'clock. And then I'll do a final live stream the night before the exam, which is next Thursday. So Thursday we'll do the live stream. Friday you've got your exam. Is that right? Have I got my days right there? Either way, I'll be doing a live stream the night before the exam. I hope you find this helpful. Thanks to the last few people who made it to the end. And yeah, good luck with your revision. You can check me on Instagram as well if you've got more questions. Thank you so much for watching. Bye.